Good morning. I hope you heard me well. So I am here to represent a team, basically, which is an international consortium trying to study melanoma and a very large set of sample and cases in order to find precision treatment or better optimized treatment for melanoma patient. So the work which I will present is about how we have used the Covaris accelerated acoustic sample preparation for FFPE sample preparation for untargeted proteomics, and I will speak about some of the findings that we have found based on the data generated with this workflow. So malignant cutaneous melanoma is basically only representing a small fraction of all the skin cancer, but it's the most deadly subpopulation because it's accounting for 85 to 90% of the death, which is related to skin cancer. Here you can see also the different stages, right? The early stage, generally that's if it's detected in the early phase, the success of treatment is 98%. In the second stage, when you have already metastasis to a lymph node, then it goes down to 64%, and then you have multi-organ metastasis, then the success rate is much lower, and we are also facing all type of this representation by sample that we are trying to cover to complete spectrum of the disease. So here you see some example of melanoma which can occur in the superficial skin, but it can also occur in nails, in palms, as well in other parts of the body. So our goal is to basically develop a so-called patient digital twins, which means that we try to collect all the information that a patient has, including clinical metadata, other histories. But in the center, right, we are also using tissue obtained from those melanoma sample and multiomics profiling, and together try to use all of this data as a big clinical data to find better treatment. And the digital twin here is basically representing what is essential information of the patient that allow us to tailor and optimize the best treatment for a particular patient. And this is called the Cancer Moonshot, and also the initiative is also belonging to the US started Cancer Moonshot program, and it's focusing on different aspects of melanoma. So at Lund University, who is the central core of this initiative, there's a very large biobank, which you can see here, which is automated, -80 biobank. And it contains several thousand of samples, I think 5,000 samples from around 2,000 patients currently, but it's rapidly going. And not only collecting samples from South Sweden, but also from Hungary and Mexico, and there are also discussion with other partners. So the biobank it's in academia, but working at the industrial quality level. So it means that if you have the diagnosis, the surgical treatment, you take a body part, then your sample is processed, sliced, you have fresh frozen, but most of the archive that we get is FFPE. Basically, there's also quality control and annotation, which is going in. And after that, the specimens are provided for the lab members who are performing the analysis. So we find very important to put multiomics in this big data optimization, especially proteomics, because proteins are generally the drug target and are generally the biomarkers that I measure, that the functional unit molecular unit of tissue. So we follow the individual's cancer mapping, which is part of what I have explained. And basically, you get the patient material, which is a tissue. Then we slice the tissue, and one of the slices is going to digital pathology assessment when the trained pathologist, or now we are using AI for that, is assessing different pathological scores and characteristics, and then from an adjacent slice or from the same slide that we had the digital pathology, we are generating multi-omics profile. Currently is per slice, but we are also working on laser microdissected workflows when we really go in a different part of the tissue and we don't analyze the full tissue as one. So, and we are using the obtained data, as I showed you, to find biomarker or up and down regulated pathways in which we can drug target and suggest new treatment for prolonged survival. So in the first stage of these biobank studies, for the first 500, this is the reason why it's called malignous melanoma, and 500. We generated several papers with small studies, and that's the problem that when you do work with batches, then reproducible become a key factor. Also, if you want to compare the different batches. So here we had a few papers, right, in 2024 and 25, each of them addressing different subcohort of melanoma, mostly metastatic, because that's easier to obtain than primary tissues, or assessing even immune checkpoints inhibitor or other type of targeted therapies in different subcohort of melanoma. So Lund obtained the Covaris R230 acoustic focused sample preparation, and we performed a pilot, and the results of this pilot I will present here. So melanoma, if you look at the big landscape, there are 4 known subtypes, right? And here you have also the percentage of occurrence of those subtypes. The most occurring is superficial growth melanoma, which you can see superficially growing. They are the best responders because all the treatment has been optimized to this type of melanoma. And the characteristic is they grow on the surface of the skin. There's a more aggressive form, which is 15 to 20% of the melanoma. That's called a nodular melanoma, which you can see here. They are more aggressive. They go more on deep, so the breast flow thickness is much higher. And then you have so-called lentigo malignant melanoma, which is occurring for sun-exposed sites. So if somebody is going too much to the sun, then in an old age can develop this type of melanoma. And there is one other melanoma which is developing on nails, palms, and soles, and it's not related to UV exposure. It's represent less than 5%. So most of this, I am also mentioning it because these very rare forms of melanoma generally are left out from profiling. And now in this study, we take 10 sample from each of these subclasses. And so the focus was to use, right, the Covaris high throughput focus ultrasonicator R230, which I think the benefit was already mentioned. So it's controlled. You don't have any contact with the processing. It can be used in different formats and allow automatization, including also tracing of sample, which is always a burden, right, if you have a large sample size, especially working in academia. And you can also automatize and include, in your local software environment. In this case, where we have used it for FFPE tissue, it was shown that you can also use it to other type of sample processing. So the first things that we have done was optimization, try to see which of the protocol is providing the best approach. So here we tried out. So here we tried out 4 different sample preparation protocol. You have the standard method, which is very lengthy, include many steps, and you need to use a large volume and large reagent set. And here you see, right, the concentration of protein that we get out, right, using this standard method, which will be the reference. And here you see a protocol that uses Covaris, the paraffinization. Here, without the paraffinization, SDS buffer. And here, one-step deparaffinization with Covaris buffer. And here, one-step deparaffinization with SDS. So these dashed columns are related to glass vial, which is very hard to automatize, and that's why it takes a lot of time to analyze one sample with Covaris, right? The processing time is much lower. And through we get higher concentration for the glass vial, but the concentration that you get here with the Covaris was sufficiently enough quality to enable high throughput profiling. This is shown by the number of identified protein, right, which goes to until 5,000 protein in one sample. So for the Covaris, and if you compare that with the standard method, it doesn't reach the 5,000. So we get a slightly better number of identified proteins. So when you look at the reproducibility, so coefficient of variation, you can see that the standard method has relatively large variability. The method, right, with the first 2 buffers also provided quite large of reproducibility, but the one-step deparaffinization with Covaris buffer or Envision lab, we have provided better reproducibility. So this is for peptide, and of course you infer the proteins from the peptide, so you see basically the same picture. We also optimized the length of the sample preparation with the 2 methods, so with using Covaris buffer and SDS buffer after the paraffinization, and we basically use then the one-step paraffinization with SDS. And we also checked that at 5 minutes you already get most of your proteins. So basically we ended up with 5 minutes of sample preparation. So here you see the workflow, right? We get one sample, you do a cryotrom, so those are FFPE tissue. We take 2 slices. One slice goes to digital pathology, and the other slice is going through the proteomics workflow with one-step deparaffinization, then several cycles of using the Covaris and reverse cross-linking at almost 100 degrees. And then we do reduction alkylation and S-trap cleanup and analysis by mass spectrometry. So digital pathology and assessment of the tumor is very important by the pathologist. And here you see the annotation that this type of workflow allows. And basically, it's also allowed to assess which type of profile we get for proteomics, and also allowed to include that in the statistical analysis. So here you have the 4 subtypes. Here you have the lentigo, malignant melanoma, right? This is skin exposed, and you can see here with the digital pathology and pathology annotation that the characteristic of cellular elastosis is characteristic of lentigo malignant melanoma, and which is represented here at a large surface. As well, adnexal involvement is included. For superficial spreading melanoma, right, you have mostly a tumor dermis with some sign of regression, very small adnexal involvement, which is not characteristic of this type of melanoma, but can occurring. And you can see also the activity of tumor-infiltrated lymphocytes. For nodular melanoma, you can see that the depthness is much higher of the tumor content. You even have necrosis because the cells are growing very fast, and this type of nodule is characteristics of the aggressive form of nodular melanoma. And here you have this rare form of acral lentiginous melanoma, generally characterized by this very thick stratum corneum. And basically, you have also a lot of junctional tumor between epidermis and dermis. We have then performed differential expression analysis using lasso and logistic regression. And here you can see the differential compounds between, here I put the two aggressive melanoma, nodular and artigo, acral lentigo melanoma. And here you can find all the enrichment, right, which is characteristics, here the clinical parameter as well. You can see that both the nodular and the ALM has more thickness, more thicker cancer. The type of the progression is more advanced, also they are more aggressive, and ulceration of the melanoma is also more frequent in these two types. So there was not many information about the acral lentiginous melanoma. This is the rarest form, and this is the reason why we focused more on assessing how much information we get for accurate treatment, especially because it's a non-UV exposed melanoma. And here you see some of the markers that are upregulated in this acral lentiginous melanoma. You see here 4 genes, 2 genes, T and C and post-N, are related to extracellular matrix remodeling and microenvironment remodeling, and post-N is only upregulated to this acral melanoma. For RF4, they are responsible for membrane trafficking and integrin recycling, and EF4, it's basically related to proliferation and tissue invasion. So if it's upregulated, then the tumor is more aggressive. So we then take ALM, and we did differential analysis of against all the other subtypes. And you see here all the upregulated pathways and the downregulated pathways. So I want just to emphasize the upregulated extracellular matrix reorganization, which is needed when you have an aggressive form of the tumor to grow, and also the start initiation and translation elongation, right, to build up the proteins. So protein synthesis is going up in this type of melanoma. What is going down is lipid metabolism, fatty acid biosynthesis, branch chain amino acid catabolism, and chromatin remodeling is going down in acral lentiginous melanoma. So finally, we have also seen one protein going down, right? This was the ETP5 inhibitor factor 1 with respect of all the other melanoma, and this factor is acting in mitochondria complex 4 effect. And first we wanted to see if it's also related to the number of mitochondria. You can see that here you see a few markers which is related to the number of mitochondria. This was more or less the same for all the subtypes. The number of complex 4, this was also didn't really change, and also for the dynamics, how mitochondria is remodeled and changed in the cancer process. So we was able also to include those factors in the differential analysis to correct for those. You can see that none of these factors is basically affecting the fact that this inhibitor is going down. The other aspect was to get markers for glycolysis per normalized per complex 5. Complex 5 is responsible for oxidative phosphorylation to see that basically glycolysis in acral lentiginous melanoma and also in nodular melanoma is higher compared to the other two subtypes. Why this is important? Because if you have a mitochondria, you have a hydrogen pump. Here you have, this is complex 4, right, with 2 subunits. This is the normal functioning when you have normal function of oxidative phosphorylation, and you have ATP production, which is the most efficient ATP production related also when you have sufficient oxygen. And basically when you have hypoxia or other type of stress, this proton pumping is reversed, but in order to maintain the intact form of the mitochondria, right, this inhibitor should act and should prevent that the cells is decomposing ATP and basically die. But since this is downregulated, this protective effect is not there. And this is the reason why this type of cancer is related to glycolysis, which was often shown here, and lead to Warburg effect. Right? So this is here, you can see also the structure of complex 4, and this inhibitor is acting at this subunit. So for take-home message, right, so for us, the Covaris R230 allows high-throughput automatic and parallel analysis of large, here it was still a pilot, but allow analysis of FFPE tissue archive. We have identified, even in this small pilot, a key inhibitory regulator for Warburg effect in acral lentiginous melanoma. And now we are scaling up the workflow to analyze all of our sample, which is more than 5,000 samples obtained from different countries. And basically, we will use this data, right, to build digital twins and to provide precision treatment or optimized treatment for melanoma patients. If you want to hear more about this story, then please come on Thursday to poster 607 in the poster time, and we can have a more deep discussion. Thank you. And maybe also the affluent legend. So here you can see Jerry Marco Varga, who is leading all this initiative, and also Yamanis Gilles, who initiated all this project, or the Lund team as well, other members from Hungary, or from me from University of Groningen, and all the supporters for the project. So thank you very much for your attention.